Molecular oncology and the use of biomarkers is becoming key for the understanding of how the tumor behaves and also the drugs that we use. So, for example, we need to understand better how heterogeneous is the tumor, how the tumor evolves during time. So we use biomarkers to understand the prognosis, the tumor evolution, and in terms of how we use therapies, biomarkers are key for the use of targeted therapies and immuno-oncology. For targeted therapies, for example, those are therapies that are hitting a specific signaling pathway. So we need to understand how hard are we hitting the target. Those are what we call pharmacodynamic markers to understand if the drug is doing its work, that is its purpose. And then for immuno-oncology, we need to understand how the host, the patient, immune system is behaving, and also how the tumor is defending from this attack of the immune system. Main limitations of assays used in the clinic is the degree of validation that gets when they are used in clinical trials, because it's not the same validation that you get in the lab. So occasionally we get assays that need more stringent criteria to be used in the clinic. On the other hand, another of the limitations is the number of samples that you can get in the clinic is limited. And the third one is probably related to the different clinical trials and how to use information from the same assay across clinical trials and across companies. For example, in the area of immuno-oncology, we are very interested in understanding how the tumor reacts against the immune system after a checkpoint inhibitor. But we have several clinical trials and several companies, and it's difficult to aggregate all this information. I think there are two different issues. One is the stress that we have put on preserving confidentiality. I think as physicians, we are used to preserve the confidentiality of patients or of samples. But I think the regulatory environment has stressed and overwhelmed the industry and academics with this issue a little bit too much and complicates things. The other issue is the possibility of combining or aggregating data from different clinical trials, especially when they are from different companies. This is a major hurdle to really understand better some of the mechanisms of resistance. It's important to coordinate the efforts of pharma and academia, because they are coming from two different worlds. Pharma companies normally need to predefine what kind of analysis, especially for budget purposes, but also occasionally for regulatory purposes. Academicians behave very differently. We react quickly to emergent information in the literature or maybe some other internal data. But it gets very complicated to influence the use of biomarkers when a clinical trial is ongoing, for example. Public databases are becoming fundamental. We are in the era of big data. We are in the era of stratified medicine, so looking for developing drugs for small patient populations. For that, we need to join efforts. No single institution can do good clinical research nowadays. It's all collaborative research, but it's going even beyond networks of academicians. We need public databases because there is a lot of great matter out there that can use this information. So having all this information in the public domain also helps to the reanalysis of this information. And occasionally you can have very important findings when somebody with different eyes look at the same information. That's why having available data in public databases is key.